Hi, my name is Olivia, and I did my independent study this semester on the immune system and immunotherapies. So, this is just going to be a brief overview of the main topics that I covered. So, first off, what even is the immune system? For a quick overview, the immune system is a collection of cells, tissues, and molecules that are involved in immunity reactions, which is basically your body's response after it's exposed to a virus or some sort of pathogen. The most important physiological function of the immune system is to prevent and eradicate infections. So, the immune response is the coordinated response of those cells, tissues, and molecules to the pathogens your body is exposed to. And in general, the term immunity refers to protection against infectious pathogens. So, what are the stages of host defense or your body's defense to pathogens? So, there's two main types of immunity. There's innate immunity and adaptive immunity, and there's a few key differences between them. So, innate immunity is always present in healthy people to block the entry of and eliminating pathogens. So, it's constantly working to defend against infections. It's the first line of the defense. For example, the epithelial barriers of your skin and your mucosal tissues that produce antimicrobial substances to protect you. And it will eventually initiate your adaptive immune response. The one kind of drawback is that since it's so generalized throughout your body, there's a limited diversity. Um, So it responds in ways that are also characteristic of adaptive immunity, but much more rapidly and limited diversity of antigen recognition. So this is in contrast to adaptive immunity, which activates in response to the proliferation or just like generation of lymphocytes, which are a certain kind of cell. And it takes a few days to develop, and it kind of evolved as a body's natural response later in evolution. There's two types of adaptive immunity that are distinct by being mediated by different cells in different locations. So there's humoral immunity, which refers to antibodies that are produced by B lymphocytes, which is a certain type of cell, to eliminate extracellular microbes, so just pathogens outside of the cell. And that prevents microbes from infecting tissue cells and neutralize toxins. And then there's cell mediated immunity, which focuses on T lymphocytes, a different kind of cell, to eliminate microbes inside the host cells. So basically, outside versus inside immunity cells. This is just a quick graph that shows kind of like where your innate immune response is activated versus, which is right at the beginning here, versus your adaptive response across time and the proliferation of different cells of T cells and antibodies. So T cells is your, as we said, your cell-mediated immunity, and antibodies is your humoral immunity. And then these are just the different types of cells in innate immunity versus adaptive immunity. So some of the key tissues around adaptive immunity, which I focused on more, is the primary lymphoid organs and tissues. So that's where your B and T cells form, multiply, mature. So, for example, in the bone marrow or the thymus. And then there's the key secondary lymphoid organs and tissues. So that promotes the development of adaptive immune responses. It brings together T and B cells that are specific for the same antigen. And two key secondary lymphoid tissues are the lymph nodes. So when you have lymph fluid constantly leaking out of blood vessels to pass through, and eventually it comes back into blood circulation. It also stores sample antigens for APCs, which are antigen presenting cells. It's a certain type of cell that activates the adaptive immune system. And then the spleen is also the other key secondary lymphoid tissue. So that's in your abdomen, and it's for more blood-borne antigens. There's like a separation between red and white pulp. So the white pulp is the lymphoid region that activates the adaptive immune system. So some of the key cells of adaptive immunity. So there's lymphoid cells, lymphocytes, which I mentioned before, and those mature from cells in generative lymphoid organs. So those primary organs that I talked about, the bone marrow and the thymus, and then from there they move and are activated at the secondary lymphoid organs. There's 3 cells that I'll just focus on for an overview. There's obviously a lot more in the adaptive immune system. The first, which is pretty key, are T cells. So there's 2 kinds of T cells. There's CD4 plus helper T cells, and that helps produce B cells to produce antibodies and phagocytes that destroy microbes. And then there's the CD8 should be minus T cells, which are cytotoxic T cells, and those kill cells with microbes. The second is B cells. So those produce membrane-bound antibodies that recognize antigens and activate those cells. And then there's APCs, antigen-presenting cells. So those are located at epithelial barriers, for example, like your skin. They capture and then they present the antigens to lymphocytes. So they kind of act as a transporter moving from your first line of defense from innate immunity, let's say, at your epithelial barrier, and then kind of giving that message to your body to presenting those antigens to initiate the rest of your immune response. So, for example, here's a mechanism of an antigen presenting cell working to activate the adaptive immune response. Okay, so what is a typical immune response and how does that look like? So this is a lot of information, but I'm just going to summarize it in general. First, we have our innate immunity at the epithelial barriers, which are the first line of defense, and that's just where your skin or your body interacts with the external environment, and that will stimulate the innate immunity, which stimulates inflammation and antiviral defense mechanisms. Then you have the APCs that will stimulate your adaptive immunity, which leads to your humoral immunity activation. So those are your B cells that secrete antibodies, and then also your naive T cells that will differentiate into effector cells, which are just more activated cells. They kind of proliferate and generate more and more T cells using certain cytokines, which are just things that are like chemicals or things that are secreted, for example, IL-2. And then once your body has kind of fought off the infection, there's the decline of the immune response. So cells are then deprived of those like survival signals like IL-2. That leads to death of the most of the cells, and your immune response subsides within 1 to 2 weeks after infection is eradicated. After that, only memory cells remain, which are a specific type of cell that can survive in your body for long periods without antigens, and that allows you to have more effective responses to the repeated exposure of the same antigen. So when you encounter, let's say, the same virus again, your body has a memory of how to fight that off and can have a more effective response. So how is your T cell response regulated? Because we can't just have cells being generated and generated and generated because that leads to cancer. So there's checkpoint blockades. So your immune responses are regulated by both activating receptors, for example, TCR complex and CD28, as well as inhibitory receptors, so CTLA4 and programmed cell death protein 1, PD1. So co-inhibitors suppress the activation of T cells by blocking and removing B7 molecules from the surface of APCs, and PD1 limits responses to infections. Okay, so how does cancer develop from this? So cancer, as I'm sure most people are familiar with, is a disease in which abnormal cells divide uncontrollably and destroy body tissue. And so cancer has evolved certain mechanisms to be able to escape your immune response and immune surveillance. So for example, just a few, it creates defects in antigen presentation to slow your adaptive immune response. It can hijack normal mechanisms of immune checkpoint control, So like CTLA4 and PD1 that I described in the last slide, by turning those off, and then also induces T cell exhaustion, which basically means your T cells eventually might be effective at the beginning, and then they kind of get less and less effective. So what are some of the traditional cancer treatments that are not immunotherapies? There's chemotherapy, radiation therapy, bone marrow transplant, um, and cold needle cyroprobe. And some of the harmful side effects, though, of these are that it leads to increased risk of infections due to immunosuppression. So your immune system is heavily suppressed to prevent the treatment from damaging healthy cells. So if you do happen to encounter a different virus while you're under that immunosuppression, you're a lot more susceptible. Extreme pain and fatigue. You can have hair, skin, and nail problems because you are sometimes losing those healthy cells. And can be pretty ineffective. It's usually 60-70%, but it really ranges based on the cancer and the type of treatment you're getting. So what are immunotherapies? So immunotherapies in general are just a very exciting new field of research in the immunology world. And there's couple different types though. So there's first oncolytic virus therapies. This is kind of the first initial form of immunotherapy that was discovered when William Coley noticed that mixtures of live and inactivated combinations of those strains of bacteria cause tumor regression in sarcoma. And so oncolytic virus therapies leverage genetically modified viruses to infect tumor cells, so that ends up stimulating an inflammatory environment and invoking like your immunities. And that's an example of a genetically modified virus that's used. And this is just a diagram again explaining how oncolytic virus therapies can be effective. There's also cancer vaccines, which, as it sounds, is pretty self-explanatory. It utilizes tumor-specific antigens to trigger your T-cell mediated anti-tumor immune response. So those antigens are then transported to the lymph nodes, which is one of the secondary lymphoid organs, and that activates your adaptive immune response. We also have cytokine therapies. So cytokines, those are the things that are secreted by antibodies and other kinds of cells. So activated T cells naturally produce the cytokine IL-2. As I said before, that stimulates the proliferation of T cells, called clonal expansion, so just expanding more and more T cells. So when you have the synthetic administration of large doses of IL-2 or other interferons, that can also help stimulate T cells by generating more T cells to fight the cancer. However, this can also lead to poor tolerability and severe toxicity effects because you're not having as much regulation. Okay, and then there's also immune checkpoint inhibitors. So we covered how the T cell response is regulated by inhibitors to maintain an immune tolerance, but those inhibitors with cancer cells are usually used to evade immunosurveillance. So ICIs, immune checkpoint inhibitors, interrupt those co-inhibitory signaling pathways to kind of reinvigorate the anti-tumor immune responses. So it's blocking CTLA4 and PD-L1. So that could induce more effective immune responses because you're not having that regulation that's suppressing the T cell response, and it's reinvigorating the cytotoxic ability of T cells. However, because you aren't regulating your T cell response, there's a higher chance of developing some autoimmune disorders, which is where your T cells attack your own healthy cells. So this is, again, just a diagram of how immune checkpoint inhibitors work to block inhibitory checkpoints. And then what I also kind of focused on when looking at research papers was mainly adoptive cell transfer, which is better known as CAR T. So CAR T therapy utilizes isolated genetically engineered immune cells, so usually T cells. So it takes those and isolates those T cells, and then it expands immune cells ex vivo. So it takes them outside of the body. It engineers them a bit, which I'll get into, and then reinfuses them back into patients to be more effective at eliminating cancer cells. So chimeric antigen receptors, also better known as CAR T cells, and TCR engineered T cells have advanced in the treatment of malignant tumors. So CAR T in general, it uses the antibody fragments specific for the cancer to recognize specific antigens on the surface of cancer cells. And it really advanced in 1998 with the addition of some co-stimulatory molecules such as CD28 into the engineered CARs to allow T cells to remain active and effective. So, what are chimeric antigen receptors or CARs? How are they structured? So, CARs are engineered synthetic receptors that function to redirect lymphocytes, usually T cells, to recognize and eliminate cancer cells with that specific target antigen. And so, there's four main components of CARs that can be engineered. There's the extracellular target antigen binding domain. So that is the portion of CAR that's specific for your target antigen. So that'll be different for each tumor, for example, for different cancers, because there will be different antigens, target antigens for each of those. There's also the hinge region. So that just extends the units from the transmembrane domain to the able to reach the antigen on the cell. Transmembrane domain, it's the least characterized region and to anchor, and then there's the intracellular signaling domains, for example, CD8, CD28, and so those are some of those co-stimulating molecules that help the T cells remain active. And CAR T has been a very exciting advancements. There's been about 7, I believe, at this moment, CAR T therapies that have been approved by the FDA. It's been shown to be very effective, almost 99 to 100% in some cases, for certain cancers, especially blood cancers. But with that, there also comes some limitations that scientists are still working through. One of those is antigen escape, which is when your tumor resists to the single antigen targeting CAR construct, which in simpler terms means that sometimes mutations will cause the antigens on the tumor cell to disappear, and then the CAR T cell is pretty non-functional at that point and non-effective. There's also on-target off-tumor effects. So that's when your target antigen might be expressed on your normal tissues, which means while it's on target for the antigen, it's targeting cells that are not on the tumor, which can cause autoimmune disorders. There's also CAR T cell trafficking and tumor infiltration, which is just how effective you are actually getting your T cells back to the tumor in a rapid manner. And so something they've been working on that I read about in my papers is local delivery directly into the body at the tumor location. And then a big issue is neurotoxicity and cytokine release syndrome, or CRS. And so again, that's just more toxic side effects to the body as a result of CAR T therapy. But that's everything kind of I covered this semester around immunology, the immune system, and immunotherapies. So thank you for listening.